Hello, my name is Olivia Rivellini, and today I will be describing my research with the Cullen Lab on determining the effects of neuronal proximity to blood vessels on cell membrane permeability following repetitive traumatic brain injuries. Our lab studies the pathophysiology of brain injuries. My work involved determining if these injury outcomes are influenced by different anatomical structures in the brain. Traumatic brain injury, or TBI, is an external force that results in brain dysfunction or damage. We are specifically interested in inertial loading TBI when the brain is accelerated or decelerated within the skull. Repetitive TBI is especially damaging to cerebral tissue and vasculature. Looking at the pathology of this type of injury, we are concerned with blood vessels and how their presence in the brain may cause different injury patterns in the brain tissue. Because blood vessels have different densities than the brain parenchyma, we believe these structural discontinuities create weak points in the brain, which make the junctions between vessels and neurons more susceptible to damage. We hypothesized that neuronal vulnerability to cell damage, or cell permeability, following repetitive TBI is a function of proximity to mid-sized blood vessels. To test this, we had to define parameters for what it means to be proximal to a blood vessel. We created a program in an imaging software to create a digital annulus around each mid-sized blood vessel with a set diameter of 200 microns and called this area the perivascular domain. Within the annulus, we have 3 different color zones to illustrate different degrees of proximity to the vessel. So the red zone is 0 to 50 microns away, the blue is 50 to 125 microns, and the green zone represents 125 to 200 microns away from the vessel. The experiment was conducted by first infusing a lucifer yellow dye marker into brain ventricles to identify cell permeabilization after the injuries occur. The first group of pigs received 2 injuries with 15 minutes in between, and the second group received 2 injuries with 3 days in between. The inertial loading injury was induced in the pigs by quickly rotating their heads in the sagittal direction to mimic a closed head diffuse injury. Focusing on the midbrain regions of the porcine tissue, we used image analysis tools to identify permeabilized neurons, which you can see here in C, and to identify all mid-sized blood vessels, as you can see here in D. Our software individually measured each blood vessel and recorded those with a diameter of 25 to 115 microns. Our perivascular domain program then overlaid the color zones over each vessel and calculated cell density by dividing the number of neurons in each color zone by the area of the color zone. As you can see here how I counted each neuron, this one would be in the red zone, this would be in the blue zone, and these 3 neurons on the outside are in the green. From this counting technique and calculation, we found that the timing between repetitive injuries may have affected neuronal susceptibility around blood vessels. Firstly, we saw here that there was a positive correlation between permeabilized neuron count and increasing distance away from the vessels for both injury groups. The density calculation then revealed that for the 15 minutes between injuries group, permeabilized neuron density was greater in the non-perivascular areas of the midbrain tissue. We were then surprised to see a distinctly different result with the 3 days in between injuries group, in which permeabilized neuron density was greatest in the perivascular domain, with the density being the greatest as vessel proximity increases. So the density here was greatest in the red zone, which is the closest to the vessel, and then it decreases as it gets further away from the vessel. We then measured the sizes of the blood vessels to determine if vessel pathology was responsible for our surprising results, though we did not see any significant difference in vessel diameter between the groups. My main takeaway from this project was the novel semi-automated platform we developed to define perivascular zones and to identify blood vessels and permeabilized neurons. With this platform, I conclude that timing between repetitive injuries may be a mediating variable in neuronal susceptibility within perivascular zones. And we want to continue looking into these results with future experiments that will allow us to study the pathophysiology of TBI more in depth. Applying this research to understanding TBI pathology could inform new ways to treat repetitive TBI in the future. Thank you so much for listening.